Well, let's start our seminar today. It's a National Cancer Prevention Month, and I'm so excited to see a great friend and also colleague, Dr. David Heber. We were just talking about before pandemic, we used to see each other at least a couple of times a year, but now through the Zoom. So we are so excited to have him. And I try to summarize Dr. Heber's brief CV, and I thought he's going to go for 30 minutes. So bear with me. I'm going to go just with the outline. Dr. Heber is a professor of Emeritus of Medicine and Public Health, and David Geffen School of Medicine at UCLA. He's been known internationally prominent in the field of the nutrition and cancer, as well as obesity and age-related chronic diseases. David, as far as I know, is the very few physician who has the knowledge of nutrition in the world. So he's very well known. He's the founder of and director of UCLA Center for Human Nutrition, and founding chief of the Division of the Clinical Nutrition at UCLA in the Department of Medicine. He led more than 100 research projects with more than $100 million grants funding. He has published over 250 research papers and published 50 books, chapters, 4 books, which they are on the public interest with the best-selling books, and also national bestsellers. It is great that we talk about the one book, especially that we use for our students in the diet and cancer prevention, which is nutritional oncology. And he just a few weeks ago, he published the second edit on that. And Dr. Heber is a board certified in internal medicine and endocrinology and metabolism by American Board of the Internal Medicine, and certified physician nutrition specialist. He earned his MD at Harvard and his PhD in physiology at the University of California at Los Angeles. He has been recognized as the best doctor in America several times. Dr. Heber's primary area of research is role of nutrition, phytonutrients, botanical dietary supplement in prevention and treatment of the several types of cancer, obesity, cardiovascular disease. And beside of all of these things, he's a great speaker, great mentor, and I have known several of his mentees that they are very successful in nutrition research. So having that, I would like to welcome you, David, and good to see you.
Thank you so much, Reza. It's a wonderful invitation, and I'm so happy to be with all of you today to discuss the topic of nutrition and cancer. And I'm going to go ahead and share my screen and leave some time for discussion at the end. So let me start here. Let's see, slideshow. There we go. So you should be able to see my slides now. And I think the interest of the National Cancer Institute in cancer control initially focused on early detection and treatment, and less attention was focused on prevention, with the exception of research on tobacco and cancer, which was pioneered by the late Dr. Ernst Winder. Then in the late 1960s and 1970s, this began to change, as researchers saw a growing number of occupational, environmental, and lifestyle factors that affected cancer. And Congress and advocacy groups, including the American Cancer Society, pressured the National Cancer Institute to devote more resources to studies of cancer causation, nutrition, and primary prevention. And this resulted in the NCI-designated comprehensive cancer centers around the country. Research on diet, nutrition, and cancer increased as part of the war on cancer following the 1971 Cancer Act and the formation of these cancer centers. And federal support for diet and nutrition research was increased as a result of the growing public interest in prevention, while skepticism remained among cancer researchers and clinicians. Now, I just recently, as Dr. Reza mentioned, I just recently published this book, which is a textbook together with Dr. Xiaoping Li, who's the current director of our center, and Dr. Bill Goh. It's on nutrition in cancer prevention, treatment, and survivorship. And the role nutrition plays now in cancer causation, treatment, and survivorship has received increasing interest with the development of immunotherapy and genetics for precision oncology. So this is leading to an active area of research by scientists and physicians, but still engenders considerable controversy in the oncology community and among many basic scientists. Within the cancer research clinical community, nutrition has traditionally been considered a side issue. But through a lot of hard work over the last 20 years or so, there's been a lot of increased interest in nutrition. And over the last 15 years, the increased use of genotyping of cancer specimens using next generation sequencing has led to the development of therapies that take advantage of the amplified metabolic pathways and surface receptors for growth factors in cancer, many of which are affected by nutrition. So if we go back to the early epidemiological work of Richard Dahl and Richard Pito, this is back in 1981, they published surveys showing that, estimating that 35% of all cancers were due to diet. A diet rich in fruits and vegetables became a major area of research for many laboratories supported by the National Cancer Institute, including our own UCLA Clinical Nutrition Unit, which was funded from 1989 to 2006. In 2001, I wrote a book for the public called What Color is Your Diet? Basically emphasizing the phytochemicals in different colorful fruits and vegetables, which are both antioxidant and anti-cancer. And a lot of research in animals and some limited clinical research back this up. So if we look at diet, what does it affect when we look at this? We look at breast cancer, endometrial cancer via obesity and the estrogen receptors, the aerodigestive disease cancers such as lung cancer. All of these are through inflammation. There's a lot of evidence that smoking, of course, works together with poor nutrition in this area. Colorectal cancer, pancreatic cancer through the microbiome and through obesity, prostate cancer, gallbladder cancer, and some other cancers, all of them related to nutrition. Now, the American Institute for Cancer Research has estimated that nearly 50% of the most common cancers can be prevented by eating well, staying lean, exercising more, smoking cessation, protecting the skin from ultraviolet radiation, and interestingly, getting vaccinated. They've now developed vaccines for the human papillovirus subtypes that are pro-carcinogenic for cervical cancer. And I think now with the new mRNA vaccines that have been developed for COVID, that we will see in the future mRNA vaccines against other cancers. Now, the major risk factor for cancer is, of course, aging. As you age, your risk of cancer increases. And when you see statistics on cancer, you're always going to see them as age-adjusted risk, because the Tomasetti and Vogelstein proposed that a significant portion of human cancers were simply due to bad luck. Bert Vogelstein at Johns Hopkins was the one who had developed this idea that all cancers went through an orderly progression of genetic changes. And he developed information on a number of what they call oncogenes, mutated normal genes that promote cancer growth. And he felt that these were cancers that occurred in cells that were rapidly turning over, so that bad luck statistically drove somatic cell mutation events during DNA replication, and only a few of these led to carcinogenesis, or inherited genes that could impact these public health policies. Well, Albini and co-workers who were at the National Cancer Institute at the time pointed to an alternative explanation of the association of cancer and aging called the cancer stem cell hypothesis. So stem cells are undifferentiated cells that can then be geared to become all of the various organs in your body with the basic blueprint of the DNA that you have in your genome. So this concept argues that cancers are derived from tissue stem cells and not from somatic differentiated cells, and emphasizes the importance of the tissue microenvironment in the growth of transformed cells. Since aging is inevitable, the statistical mutation model supposes a helplessness in avoiding cancers, whereas extensive knowledge of known risk factors and behaviors that can lower the risk for specific cancers, including smoking cessation and changes in diet and lifestyle, indicate that prevention should still be a primary goal for public health policies. So instead of just passively waiting for a cancer to be diagnosed and then have some chemotherapy prescribed, the idea is to take an active role in smoking cessation, diet, and lifestyle to prevent cancer. Now, the primary hypothesis of aging is one that was brought up by Dr. Caleb Finch. We called him Tuck Finch at the University of Southern California, and he called this process inflammaging, that inflammation with aging occurs. Now, as cells age, they either die after a certain number of divisions, or they form what's called a senescent phenotype. The senescent phenotype is characterized by the secretion of a lot of cytokines, which cause DNA damage, impaired immune function, oxidative stress, and so that inflammaging and inflammation promoted by obesity, poor diet, smoking, and stress is one of the primary factors for both aging and cancer. Now, another thing that happens in our society is people stop exercising, and physical inactivity leads to a loss of muscle mass. And this can happen in non-obese people or obese people, but it leads to an increase in body fat because muscle accounts for the majority of calories that you burn at rest. So as your muscle mass goes down, even if you're eating the same number of calories, your body fat will go up. And adipocytes, the body fat cells, especially in the abdomen, are pro-inflammatory. So they play a major role in this process. In addition to that, within the muscle, the mitochondria, the energy factories in the muscle, with aging, develop a leak of oxygen radicals. Exercise can help to prevent this and lead to healthy age muscle, whereas unhealthy age muscle can lead to such degree of sarcopenia that by age 60, many people are disabled. So diet and cancer prevention has many different aspects as outlined here by the World Cancer Research Fund and the American Institute of Cancer Research. Limiting the consumption of red and processed meat, limiting the consumption of sugar-sweetened drinks, limiting alcohol consumption, limiting consumption of fast foods and other processed foods high in fat, salt, and sugar, eating a diet rich in whole grains, colorful fruits and vegetables, and beans, being physically active and maintaining a healthy weight. And for mothers, breastfeeding their babies. We have a lot of data now that breastfeeding sets up the immune system and the microbiome early in life and leads to positive epigenetic changes. And there are many things carried in the breast milk, including protective antibodies and DNA information from the mother. And after a cancer diagnosis, there are many survivors alive today, and you want to follow the same recommendations during survivorship. Now, basic and translational cancer research has really advanced in the last few decades. So what scientists do is they take patient-derived cell lines, culture them, implant them in immunocompetent mice, and these xenograft models can then be studied to find certain therapies that may work. And that is called targeted cancer therapy. One of the biggest positives on this is the development of imatinib, which is known as Gleevec for chronic myelogenous leukemia and gastrointestinal stromal tumors. They have totally revolutionized survival in this particular cancer. And there are other examples of targeted therapies that are very, very good, and it can be used together with early diagnosis to help people live longer than ever before. Now, the genotyping of breast cancer has led to the definition of different types of cancer based on studies of the cancer cells. So there's a luminal A type, which is estrogen receptor positive and or progesterone receptor positive and HER2/neu negative. This is the most differentiated form of breast cancer, commonly occurs in postmenopausal women, and is very much related to obesity after the age of 50. The double negative, HER2/neu positive, negative cancers are very aggressive and metastatic disease. Even with these, there have been some recent developments of targeted therapies that affect the glucose pathway in these cells that have shown very positive results. Now, the one area where there's been really revolutionary results has been metastatic melanoma. Melanoma occurs from exposure to ultraviolet light, usually from sun tanning, especially in people who are light complected and don't have much melanin in their skin. There's an absolute epidemic of this in Australia because Australians are immigrants from England, which is in a northern environment, and they actually have a pigment in their skin that amplifies ultraviolet light rather than quenches it. And they do get various kinds of mutations that promote cancer. And these, even when they are very widespread, can be countered by immunotherapy, and we'll talk more about that. So precision oncology looks at the genetics for precision nutrition, and genetics may also help us understand personalized nutrition based both on phenotype and genotype, which involves the metabolome as well as the genetics, epigenetics, and body mass index, what people are eating and so forth. This promises a new era in the evolution of nutrition research and cancer prevention, treatment, and survivorship. So it's a gene-environment interaction. And genetics has uncovered heterogeneity, even within tumors within the same organ. And the discovery of imatinib or Gleevec and other tyrosine kinase inhibitors has revolutionized cancer therapy for leukemias and gastrointestinal stromal tumors. And advances in immunotherapy have been based on the fact that tumors secrete a protein which blinds the immune system to the tumor. And if you infused these people with an antibody to that protein, now the immune system can see the cancer, and even when it's widely spread, it turns out it can eradicate metastatic melanoma and lead to long-term remission. And what they have found is that the microbiome is very much involved in this, because 70% of your immune system is located around the gastrointestinal tract. And what they have found is that certain patients don't have as durable a response. They have a different microbiome than those who do have a durable response. So what makes a cancer? Well, the most normal cells live and die in a normal cycle. However, many of the cancer cells avoid immune destruction by putting out these proteins. They deregulate cellular energetics by stealing substrates from normal cells. They have genomic instability and mutation, and they even in their microenvironment have tumor-promoting inflammation. Now, they evade immune destruction by releasing immunosuppressive factors and these T-cell checkpoint dysregulation, where they put out proteins that inhibit the ability of the T-cell to see the tumor and kill it. Now, immunotherapy, there are several methods of immunotherapy. One of these is therapeutic vaccination, which I mentioned for human papillomavirus, but we also have these checkpoint inhibitors, which are antibodies that inhibit the protein that is being secreted by the tumor to blind the immune system. And then another method is something called adoptive cell transfer, where you take the patient's own lymphocytes and you create a chimeric white cell that contains some of the tumor-specific antigen, and then you reinfuse those cells into the patient and they target the tumor and kill it. Now, the checkpoint inhibitors are demonstrated here. The tumor cells bind to T cells to inactivate them with this protein. However, if you have the block, you can now change that with a drug, and then that allows the tumor to be killed. And the two types of this are the anti-PD-1 and the anti-PD-L1. And not all tumors produce this PD-1. So you have to check for that to see that the tumor is actually producing this. And when it is producing this, you can get a good result with these types of therapies. But it also requires an active immune system from the GI tract. So there's exciting and emerging science supporting the notion that the microbiome, the 14 trillion bacteria in your gut, which are adjacent to the immune system in the wall of the gut, can affect immunotherapy in advanced cancers with checkpoint inhibitors. In patients with metastatic melanoma treated with the anti-CTLA-4 antibody ipilimumab, treatment with broad-spectrum antibiotics impacted the gut microbiome and was associated with a poorer response, while particular bacterial populations within the microbiomes were associated with longer progression-free survival. And the way they tested this, they took those microbiota and put them into germ-free mice that had a tumor implanted, and they recapitulated the observation that certain bacteria promoted immunotherapy and others did not. So the gut microbiome in melanoma patients was associated with response to treatment, also with anti-PD-1 antibodies. In patients with advanced cancers treated with immunotherapy, resistance was associated with an abnormal gut microbiome and with broad-spectrum antibiotic therapy. In support of these observations, fecal transplants from responders into germ-free mice enhanced anti-tumor immunity to xenograft tumors compared to patients who received broad-spectrum antibiotics and their microbiome. So here's a picture of this to further explain it. So if you take a fecal microbiota transplant into these germ-free mice from somebody with a non-responder, the tumor size is larger and the immune activation of the T cells is less. Whereas if you take the fecal microbiota transplant here, the tumor size is low in the responder and the immune activation is greater, showing that the microbiota definitely play a role in the response to anti-PD-1 therapy. And this is a really important observation. By the way, in 2017, the Nobel Prize was awarded to Dr. Jim Allison, who is at MD Anderson for the discovery of immunotherapy in cancer. Now, the tumors also have their own microbiome. So the blood vessels within the poorly perfused areas of tumors are able to permit bacteria to settle in areas where the immune cells cannot clear them easily, enabling bacterial growth. Anaerobic and necrotic areas of tumor tissue promote the growth of anaerobic bacteria. Each tumor type has a distinct microbiome composition, and the intratumor bacteria are mostly intracellular and present in both cancer and immune cells. The bacteria persist both in metastatic lesions and in tumor xenografts in mice. Treatment of mice bearing colorectal cancer xenografts with metronidazole, which is Flagyl antibiotic, decreases tumor bacterial load and tumor growth, suggesting the bacteria may have a role in tumor proliferation. These observations on the role of the microbiome in cancer suggest a new pathway through which nutrition, including prebiotics and probiotics, by altering the microbiome, may be used to enhance immunotherapy by modifying the tumor microbiome as well as the gut microbiome. So now let's look at other aspects of cancer. Normal cells versus cancerous cells. There's hyperplasia, dysplasia, and cancer. If we can catch these early and remove them, as we do with colon polyps, this is another way to prevent cancer. So most normal cells follow a steady cycle of cell growth and death as normal cells replace lost cells. The cellular and molecular lesions secondary to aging that promote an increased risk of cancer can be genetic, including mutations, deletions, or translocations, or there can be epigenetic changes associated with cancer. Dr. Stephen Horvath at UCLA has developed an epigenetic clock, and he finds that methylation of the CpG islands in DNA increases in somatic cells as you age, and in cancer cells, it's even greater. So both DNA methylation and histone modification occur over time, and global DNA methylations accumulate with aging and directly contribute to cell transformation. Other cancers are associated with decreased methylation as well, but the common link is that normal control of gene activation, which is what epigenetics does, is perturbed. So the gene code stays the same, the DNA code stays the same, but whether that gene is turned on or not is related to methylation of DNA in the promoter regions. So if we look at tumor metabolism, we go back to the work of Warburg back in the 1920s, who suggested that cancer was due to an abnormal metabolism of nutrients. And what we find today is that the tumor microenvironment has a perturbed tissue architecture, altered matrix interactions, an abnormal metabolic milieu, and abnormal blood vessels due to angiogenesis, which provide reduced nutrition to areas of the tumor. So there are a number of metabolic changes in tumors and in the microenvironment favoring growth. In both normal cells and cancer cells, carbohydrates, fats, and proteins are broken down to glucose, fatty acids, and amino acids, which enter metabolic pathways in the mitochondria to produce adenosine triphosphate, or ATP. However, in cancer cells, there's a shift in the metabolism away from energy production as ATP towards the production of substrates for cancer cell growth. And this is with increased production of NADH. Research on nutritional adjuncts for cancer treatment being explored include targeting glycolysis, the Krebs cycle, oxidative phosphorylation, glutamine metabolism, fatty acid oxidation, nucleic acid synthesis, which is historically methotrexate, lipid synthesis, and amino acid metabolism. Normal cells do not metabolize glucose to lactate when oxygen is available. Instead, they make lots of ATP through the Krebs cycle. Since oxidative phosphorylation through the TCA cycle is much more efficient, it produces 42 molecules of ATP versus only 6 through glycolysis. But as Warburg and Corey found in the 1920s, cancer cells metabolize glucose to lactate even in the absence of, or even in the presence of oxygen, and tumor slices consumed glucose and secreted lactate at a higher rate than normal tissues. And this is where the Warburg hypothesis came from. So here's the Warburg effect. You have differentiated tissues producing 36 molecules of ATP through oxidative phosphorylation. And when there's no oxygen present, glucose goes to pyruvate, goes to lactate, and only produces a small amount of ATP. In proliferative growing tissues or tumors, glucose does not go to ATP production, but instead goes to lactate and provides many of the substrates for synthesizing protein, fat, and carbohydrate constituents of growing cells. So the benefits of the Warburg effect on cancer cell proliferation is that they enter metabolic pathways to shift towards production of substrates for cancer cell growth. Research on nutritional adjuncts for cancer treatment do target some of these abnormal changes in the metabolism of proliferating cancer cells. So the functional role for the shift from energy production to substrate production is the biosynthesis of nucleotides and amino acids needed for cell assembly and growth. Branching off the linear path of conversions from glucose to pyruvate are production of ribose 5-phosphate, a precursor to nucleotide synthesis, dihydroxyacetone phosphate for lipid synthesis, and production of serine from 3-phosphoglycerate, which can be converted to cysteine and glycine amino acids for protein synthesis, and lactate from pyruvate. So these are all normal pathways that exist in normal cells, but it is the increased use of these pathways that characterizes the cancer cell's abnormal metabolism. Now, tumor oxygenation is also related to this, as you can imagine. So here we have an oxygen gradient within a tumor. As I said, these angiogenic blood vessels are not very good at providing oxygen to the tumor cells, and there are areas of the tumor that are anaerobic or necrotic. In the anaerobic areas, hypoxic tumor cells, they use glutamine and do not and provide lactate, whereas in the well oxygenated areas, there's aerobic tumor cell metabolism. So this has created a lot of controversy because some studies have shown the Warburg hypothesis being there, others have not, and they may have been studying different areas of tumor with different areas of oxygenation. Now, Warburg and Corey in the 1920s discoveries are fundamental in showing differences in the central metabolic pathways operating in malignant tissue. While they proposed their theory as the explanation for tumorigenesis, the identification of gene alterations, including oncogenes and tumor suppressor genes in the 1970s by Bishop at University of California, San Francisco, for which he received the Nobel Prize, he actually showed that these were normal genes that were mutated in cancer and promoted a multi-step process of carcinogenesis. All of this genetic emphasis and actually the identification of the genome reduced interest in nutrition and metabolism among cancer researchers. However, as proteomic and metabolomic research has progressed, there are nutrition research opportunities in the area of cancer metabolism. Various subcellular signaling pathways amplified in cancer cells secondary to genetic mutations favor the growth of cancer cells in the microenvironment where these cells interact with stromal cells. This year, I read an article in Science where they looked at some of the protein-protein interactions in cancer that were quite distinctive. Some of these alterations may be susceptible to inhibition, but the general interest in treating metabolic targets as a research area is still far less prevalent than precision oncology based on cancer genetics and more recently immunotherapy. Now, stromal cells are very critical to the cancer microenvironment, and they participate through a cell-cell interaction. So you can see here that not only do they provide lactate often for growth of the cancer cells, but they can also have cytokine and chemokine signaling involved, which stimulates tumor cell growth. And this has been found in prostate cancer, in breast cancer in particular. There are interactions which increase the activity of the aromatase enzyme, which produces estrogen within the breast tumor itself. So this is an exciting area of research. The problem for many cancer researchers is they study the cancer cells in isolation rather than studying them in conjunction with stromal cells or even in organoids where you reconstruct the tumor itself. Now, metabolic reprogramming of cancer cells while promoting cancer cells makes them potentially susceptible to agents that selectively starve them of their metabolic substrates. So a better understanding of cell metabolism may pave the way for that research. One of the examples is a glutaminase inhibitory drug, where glutamine is used here to enter the TCA cycle, and glutamine is a preferred substrate for normal cells under certain conditions, but even more so for cancer cells. And here's this drug CB839, which inhibits glutaminase and actually results in anti-cancer activities. So that's a new area of research. De novo lipogenesis is another one. You know, tumor cells have to make new fatty acids compared to normal cells, which depend more on dietary fatty acids because of their rapid growth rate. Conversion of acetyl-CoA to palmitate, catalyzed by fatty acid synthase, is found in cancer cells at increased levels of expression to carry out de novo lipogenesis. The free fatty acids synthesized de novo are converted into neutral lipid stores, and these lipids can in turn be released intracellularly as free fatty acids by monoacyl glycerol lipase. In patients with advanced ovarian cancer, increased monoacyl glycerol lipase activity was noted in tumors. Free fatty acids from these lipid stores can then be used by cancer cells as substrates for growth or to induce specific signaling lipids, such as ceramides, that promote cancer cell growth, including phosphatidic acid, lysophosphatidic acid, and prostaglandin E2. So de novo lipogenesis is another potentially promising target for adjunctive or therapeutic cancer treatment. Now, cholesterol is another interesting area in cancer. It turns out that hypercholesterolemia and duration of use of statin drugs is a risk factor in prostate cancer. It turns out that excess cholesterol intracellularly can be used to make androgens in prostate cancer cells, and this promotes tumor growth. Anti-androgens and substances that work even earlier inhibiting the development of cholesterol have been found to be potent anti-cancer therapies in prostate cancer. And these are very, very exciting areas where they have produced remissions in cancer patients that were not possible before. And here you see the de novo cholesterol pathway. Statins, which people take to lower cholesterol, lower HMG-CoA reductase, but there also are, there's another pathway here, feedback here from dolichols onto, and farnesyl pyrophosphate onto mevalonate reductase, and that is another way that you can attack the cancer cells' development of tumors. Now, then we're going to talk about tumor promoting inflammation as another area. Now, angiogenesis, the growth of new blood vessels, was developed by the researchers at Harvard Medical School looking at chronic inflammation in cancer. And it turns out the sustained inflammation leads to reactive oxygen species, damage to cells, and initiates carcinogenesis. Dr. William Lee at the Angiogenesis Institute and the late Dr. Judah Folkman, who was one of my professors, a very interesting man. He graduated medical school early and was the chief of pediatric surgery in Pennsylvania, and he was retrained, I'm sorry, he was of adult surgery in Pennsylvania, was retrained as a pediatric surgeon to become the chief of pediatrics at Children's Hospital in Boston, where he studied in the early 1970s the idea that tumors produce their own blood vessels to grow. And this is the area of angiogenesis. And he started his experiments with three-dimensional studies of bacteria, resigned as chief of pediatrics in order to pursue his research, and got this field to a very, very high level, and actually inhibited tumor growth with angiogenesis inhibitors. His research was not replicated by some researchers at the National Cancer Institute, and they accused him of scientific fraud. He went to the National Cancer Institute, showed them how to do the studies, and that whole issue disappeared because his research was validated. And there's a wonderful book about this called Folkman's War. Unfortunately, he died a few years ago of a heart attack while going to a conference from an airport, but he was a wonderful, wonderful professor. And this field has been very important. Angiogenesis inhibitors are in clinical trials now. So here's tumor vasculature and angiogenesis. And as you can see, there are different phases of angiogenesis. And what's really important is these angiogenesis vessels do not provide adequate nutrients to the tumor, so you end up with necrotic tumor within the tumor mass as well. You can nutritionally inhibit angiogenesis. A number of things have been studied to inhibit angiogenesis, including soy, lycopene, ellagic acid, which is found in raspberries and is a breakdown product of pomegranate ellagitannins, resveratrol from red wine. And these things are stimulated by angiogenic factors like vascular endothelial growth factor, fibroblast growth factor, and so forth. And so you can target these, or nutrition may have an effect here. Anthocyanins have also been shown to affect endothelial cell migration. And spices have been involved in vessel remodeling. So a number of areas here where you can see that nutrients of various kinds, including glucosinolates from broccoli, xanthohumol, and procyanidins, and soy, all have an effect on this angiogenesis process. So they have a primary tumor, and it cannot grow greater than about 200 microns without growing its own blood vessels. So what they do is they grow blood vessels, locally invade, then get into the circulation, and you get clinically detectable metastases. Today, they're also what we call the liquid biopsy, where you can get blood samples and look at tumor DNA at an early stage when you have early extravasation before you get to these later stages. And that's going to be a very important way to have earlier therapy. Now, there are two kinds of malnutrition. There's undernutrition, marasmus and kwashiorkor, which marasmus is what you see in a person who adequately adapts to starvation. They lose lean and mass and fat mass together. Kwashiorkor, you lose more muscle mass. This is a Swahili word for children who are separated from their breastfeeding at 1 year of age and fed cassava fruit, which is 100% carbohydrate. They end up developing edema, protein deficiency, enlarged liver, hypopigmentation, loss of hair. Overnutrition is what we really suffer with here in the United States with obesity, but around the world, you have a mix in many countries of what we call the global nutrition transition, where they've introduced Western foods into the large cities like in China and India, and in the rural areas, you still have malnutrition. So obesity still is a big factor in many forms of cancer. Adipose tissues expand and they increase the release of insulin, insulin-like growth factor, interleukin-6, leptin, which is a pro-inflammatory cytokine, and adiponectin, which comes from the lower body fat, mostly in women, is anti-inflammatory. And with obesity, the adiponectin levels fall. And what you can see is these then bind to receptors on the tumor cells, and they increase tumor growth, angiogenesis, and so forth. Cancer cachexia and anorexia remain a major problem that I've studied since 1978. This is very common. It was thought to be in 1924, it was thought to be the main way that cancer patients died. Today, we know this is a side effect of the immune system trying to clear the body of tumor cells and being unable to do so. And so now there's an increased understanding of the fuel choices of cancer cells to meet their demands for energy and for the production of biosynthetic precursors affected by intrinsic and extrinsic factors, including oncogene mutations, nutrient and gene availability, and factors in the tumor microenvironment. So there are a number of tumor effects on fat and muscle. It causes cachexia in skeletal muscle, increases lipolysis, increases brown adipose tissue thermogenesis, causes skeletal muscles to waste, and increases insulin resistance. In white adipose tissue, it causes lipolysis, but also browning. And browning of white adipose tissue is where the white adipose tissue acquires mitochondria and starts to have uncoupled production of heat. And this is a method of heat production in rodents and in young infants, but in the tumors, this can also occur. So this central nervous system has a role to play in cachexia as well. During malnutrition or starvation without cachexia, leptin levels fall and hunger increases. And if you have somebody who's liberated from a prisoner of war camp, they'll gain a kilogram a day as they really want to eat and they reduce their physical activity. In obese people, leptin levels are high, but they don't suppress food intake. But when you have starvation, leptin levels fall, and other substances like neuropeptide Y and the agouti related protein go up, and they make you eat more. So these neurons are activated while the anorexic neurons are inhibited, resulting in an increased desire for food intake. During cancer cachexia, factors such as cytokines elicit effects on energy homeostasis that mimic leptin in some respects and suppress the orexigenic ghrelin NPY agouti-related protein signaling. So the anorexia and unopposed weight loss in cachexia is the result of cytokine disruption of these central mechanisms. And I had done some research on magestrol acetate, or Megace, which is a progestational drug that increases appetite. Most of the weight gain, however, was in the fat compartment, not in the lean body mass compartment. So early research in terms of nutrition support demonstrated that many cancer patients did not gain weight despite the provision of adequate or even excess calories based on their apparent metabolic needs. While cancer patients since the 1970s have been screened for the risk of the presence of malnutrition, there have been few well-controlled trials demonstrating the benefit of nutrition on responses to therapy or survival. The area of research has receded in popularity, and the pragmatic current situation is that all cancer patients, with the exception of those at the end of life, are provided energy and substrates as required in a stepwise manner from counseling to enteral and/or parenteral nutrition. In patients with advanced cancer and malnutrition, there are a few pharmacological agents which produce weight gain and improve cachexia, such as magestrol acetate for appetite, ondansetron for nausea, and marinol or cannabis to stimulate eating, but they do not improve survival. And we have researchers at UCLA who are studying some of the unique cannabinoids in terms of stimulating eating in advanced cancer patients. So nutrition support options, if the gastrointestinal tract is functional, you should use enteral nutrition and nutrition consultation. If it is not functional, then you consider parenteral nutrition. If there is a reason to get the patient to their next level of therapy, you don't do this in end-stage patients. Unfortunately, in the 1970s, there was an industry in parenteral nutrition, and this is very expensive therapy. The patients would die and other patients would be recruited. This was the high-tech approach to nutrition and cancer. It has become less popular today, and people are more thoughtful of how they use artificial nutrition today. So now I want to talk about nutrition and survivorship. And this is a happy story because in the United States today, survivors number more than 16.9 million with projections of more than 22 million by 2030. Nearly 68% of Americans diagnosed with cancer live more than 5 years, and 18% live more than 20 years. So numerous questions arise regarding nutrition and lifestyle factors that can be implemented to maintain a healthy diet, weight, and active lifestyle, and to help prevent cancer recurrence, development of second primary cancers, and other chronic diseases. These concerns include understanding the benefits of different nutrients and dietary patterns, as well as the potential role of dietary supplements in supporting the goals of cancer survivors. Nutrition scientists are keying in on more precise advice for survivors through precision nutrition and precision oncology. So in conclusion, I want to leave you with these take-home messages. Nutrition can impact cancer prevention and treatment in the modern era. And more is understood of the molecular and metabolic resilience of cancer cells. Cancer cells exhibit oncogenic metabolic alterations, including in glutamine metabolism, the pentose phosphate pathway, and pathways for the synthesis of fatty acids and cholesterol. These cancer cell metabolic pathways produce biochemical precursors for nucleic acids, lipids, and proteins. Cancer cells achieve metabolic advantages for survival and proliferation as a result of these metabolic changes mediated by cancer-related transcription factors, including HIF1 alpha, c-MYC, and p53 linked to metabolic pathways such as the PI3K and AMPK pathway. Research on metabolic inhibitors of cancer cell growth based on genetic profiling and precision oncology are being examined in basic research and may someday yield less toxic and more cancer-specific treatments for cancer. So I'd like you to remember the following. One, cancer cells develop metabolic changes that promote their proliferation by producing substrates needed for new cells. Cancer cells interact with the tumor stroma to increase inflammation and angiogenesis. Cancer cells evade immune destruction, but immunotherapy can block this process, leading to advances in cancer therapy, even for metastatic disease. The durability of responses to immunotherapy is affected by the microbiome, leading to a potential role for nutrition in treatment and prevention. And both malnutrition and obesity, as well as age, are involved in the risks for cancer. So I'm going to stop right there, and thank you for your attention, and I'll open the floor to some questions. Thank you.
Thank you very much, Dr. Heber. That was excellent. I mean, it's overview. In the short amount of time, we learned a lot. So I opened the question. The major question is for the general public, that the effect of dietary supplements, and including vitamins, minerals, and whole host of other things in prevention of cancer. I've seen a lot of people that are taking these dietary supplements at the mega doses, which they can be harmful. Can you comment on that?
Sure. I mean, the thing that's very safe is to take a multivitamin, multimineral, which I look upon as a form of health insurance. No one eats perfectly. And these multivitamins, it's an old technology now, maybe 30 years old, from major manufacturers and even private label. They're very safe. For women, calcium and vitamin D. Postmenopausal women need 1,500 milligrams of calcium. Premenopausal need 1,200 milligrams. Men need 1,200 kilograms. Vitamin D itself is very effective and very non-toxic. And there's a lot of vitamin D deficiency because obese patients put it into their body fat. So I think, vitamin D, calcium, and a multivitamin are very good. I really believe that phytonutrient supplements do not reproduce the effects of colorful fruits and vegetables or of green tea. So I recommend people drink green tea, eat colorful fruits and vegetables. There are now dehydrated fruit and vegetable capsules, which are really nonsense because they're just have all the parts of the fruit or vegetable, and they're not really targeted. So I think we have an area for future research here. Things like curcumin, which Bart Agarwal has worked on for many years, is an interesting compound. We've done some work now in we have a highly bioavailable form of curcumin, which gets into the brain and has shown some effects against dementia. Pomegranate juice, which we've worked on quite a bit, has a lot of effects in prostate cancer. Some of these are very hard to prove because it's such a long-lasting cancer. And, I think weight reduction, exercise, these are very, very important for postmenopausal breast cancer. But individual supplements don't substitute for a poor diet. So you're absolutely right that megadoses are dangerous. And the other danger is that people get distracted from what's really important, which is a healthy diet and lifestyle.
Okay. I think we have one question. Sheena, can you read it?
Oh, how much resveratrol for prevention? I understand you need to get enough. You may need to take supplements rather than red wine and grapes. Well, it's interesting. Resveratrol is actually what's called a phytoalexin. It occurs on certain grapes that are in a humid environment. It's actually the grape protecting itself against a fungus growth. And, a lot was made of this as inhibiting, as having an effect on a histone acetylase called SIRT1, S-I-R-T. And it turned out this has not been that effective in clinical trials for cancer. So, I think this is an area where more research is needed. Okay, what is your take on the ketogenic diet approach to create an unfavorable metabolic environment for cancer cells? Well, it turns out, that cancer cells do have an increased level of glucose oxidation toward lactate. And glucose, the benefits of the ketogenic diet are by reducing very severely any sugar, both from fruits and vegetables, as well as from other processed foods. And it's so severe. It's a very rigid diet. Very few people can really maintain it. It's very high in fat. And I always recommend you use healthy fats like olive oil or avocado oil. And with that, they reduce the amount of carbohydrate down to maybe a half of what you would find in a fruit per day. So it's very hard to get your phytonutrients on that diet. However, I do believe in reducing refined carbohydrates severely. The World Health Organization recommends that women take in no more than about 6 teaspoons of sugar a day, and men about 9 teaspoons. And most Americans are way above that. They take in 20 or 30 teaspoons a day of sugar, hidden a lot of times in processed foods. Sugar is found in ketchup. It's found all over the place, even in hamburger buns. So reducing sugar and reducing starches and refined carbohydrates is a beneficial thing to do in diet, and substituting for those colorful fruits and vegetables. But I think the keto diet is a very extreme one. And actually, most people who say they're on the keto diet, they're on keto light. They can't really do it. So it's a keto-like diet. It's not a true keto diet. Mm-hmm. Do I recommend the Mediterranean diet? Yes, the Mediterranean diet is an interesting one too. You know, Artemis Simopoulos, who is from Greece, she talks about this, that the Mediterranean diet is based on observations in Greece right after World War II, when there was a lot of malnutrition in Greece. It turns out that red pasta sauce is actually a New World food. There was no tomatoes in the ancient Mediterranean diet. So the whole concept of Mediterranean diet is a wonderful marketing concept. The Mediterranean Diet Group, they love to take trips to Tuscany and drink red wine and live a very nice lifestyle. And there's been research on it in heart disease. And then there was a proposal to extend that here to the United States, and as a Predi-Med diet, it was called. And that failed because it's just not possible to do a true Mediterranean diet. So most people who think they're on a Mediterranean diet, they add a little olive oil to their salad, a little bit of pasta sauce on their spaghetti, and they think they're on the Mediterranean diet. But really, it's a diet rich in fruits and vegetables. And when I had my What Color is Your Diet book, I was invited to speak at the 30th anniversary of the Mediterranean diet in Boston. And their diet is rich in colorful fruits and vegetables, as is the DASH diet, Dietary Approaches to Stop Hypertension. So everyone agrees colorful fruits and vegetables are good for you. What role does extended fasting play in clearing senescent cells? What's very interesting, all of us fast every day. When we're sleeping, we're not eating, and there's about 40% of your genes turn off through an epigenetic effect as you change your metabolism to a starvation metabolism to use ketone bodies for energy rather than glucose, which then spares your protein. So I think the intermittent fasting is a good way to cut down on after dinner eating. Prolonged fasting is not good in the cancer patient. You will reduce your lean body mass, impair immune function, and cause a protein deficiency, which reduces responses to vaccines as well as immune function. Now, the next question, what about folate fortified cereals? Is there any evidence it poses a risk for people with high cancer risk, such as those with hyperplasia and dysplasia? Well, it's interesting. There were some studies done at Tufts University which suggested that this may be the case, that you don't want folate if you have an undiagnosed cancer because it will promote tumor growth. In the very early studies done at the Dana-Farber Cancer Institute by Dr. Farber himself, for his first study was actually on folate, and he thought folate was good for you. He would give it to children with leukemia, and a number of children died. The leukemia just blossomed when he gave the folate. And then when they gave, that's how he then got some anti-folate agents, and that's how methotrexate was discovered, and he got remissions in the leukemia. So if you have an undiagnosed cancer, folate may be a problem. We don't see a lot of reports of cancer from folate, but if someone does have a high cancer risk, it's something that should be individualized. And also, I would say there's a lot of genetic polymorphisms that are involved here in terms of methyl tetrahydrofolate reductase activity, which may be important and have an effect on choline metabolism as well. So this is a complex issue and one for research. What are your thoughts on utilizing oral nutrition supplements, Ensure, Boost, and high sugar cancer patients to help with inadequate oral intake? Well, you know, it's interesting. The Ensure and Boost are really old products. These are from the 1950s. I used to visit Mead Johnson and Ross, and they have these big you know, all the secretaries had an oak desk with a typewriter, and you had a big stack, smokestacks in the factory and stuff, and 40% fat. You're absolutely right. Today, there are other products where they have very little sugar and high protein, like 30 grams of protein and 1 gram of sugar. Those would be much healthier ways to supplement the cancer patient. And there are some that are made by Ross and by Mead. Well, I guess Mead Johnson now has been purchased by Ross. I don't know. I think Abbott makes most of these. So, and then there are some other brands on the market. But look at the nutrient profile of these supplements, because you don't want to be giving people pro-inflammatory fat like corn oil. That's not something you want in there. So that's a very old product. Okay.
Any other question? David, would you comment on the mental wellness and the cancer prevention and also treatment? Because when you are talking about Mediterranean diet, you can get all kinds of other things. But as far as I remember, they took a nap after lunch, and that was a couple of hours before they go back to work. So I do believe the mental wellness play a major role.
Absolutely. You know, stress is really, really important. We have a researcher at UCLA named Emeritus Meyer, and he's worked on the gut-brain connection. It turns out there are as many nerves in the GI tract as in the spinal cord. Mm-hmm. And stress, in their studies of irritable bowel syndrome, which is a very common disorder, actually they've shown now epigenetic changes as a result of early life adverse events. So if a child has been abused or had some kind of trauma, post-traumatic stress disorder, that can actually make changes in your genetics. And that may be a mechanism to promote carcinogenesis. So I do think definitely that mental health, adequate sleep, exercise and diet are all important. And, you know, many people don't sleep enough in our society. There was a study done at UCSF where if you got less than 5 hours of sleep per night, you had an increased risk of the common cold. So clearly stress affects immune function, stress affects the microbiome, and it can affect your epigenetic mechanisms. What do you think amino acid glutamine supplement for cancer patient with or without therapy? Well, actually, what I was saying is the opposite. Glutamine is used by the tumor, so you really want a glutaminase inhibitor, which doesn't allow the utilization of glutamine by the tumor. Now, glutamine is one of the most common amino acids in the muscle, and we use it to promote cell culture growth. So when you are growing cells in culture, that medium is supplemented with glutamine.
Any more questions for Dr. Heber? Well, Dr. Heber, we really appreciate your time.
Thank you.
And hopefully we see you here soon. Hopefully the pandemic goes away and you are going to be a part. And I know you've been in Little Rock before, but I promise you next time is going to be different when you come here.
Well, thank you, and I wish all the researchers on your team the best of luck and good research results. So thank you all for your attention. It's been a pleasure to be with you this afternoon.
Thank you. Thank you very much.